Okay, hi everyone. Welcome to the lab 11. And we will continue to talk about questions in homework 6. So in the last lab, we talked about part 1 and part 2, which focus on processing glioblastoma microarray data. And it asks you to the two parts ask you to identify two subtypes by k-means clustering, and you also perform differential expression analysis to identify differentially expressed and differentially methylated genes. And you also perform survival analysis and Cox regression. So starting from part 3, we switch to identifying mutations in tumor samples. So in part 3, question 1 and question 2, we will provide you MAF files that contain the mutations of each tumor compared to the normal DNA in patient blood. So here shows an example of such MAF files. So we will provide a bunch of MAF files. Each corresponds to a tumor sample. And if you read it in, and we are interested in the gene mutation and protein change in a mutation. So you may want to focus on just these two columns. So this file records what gene is mutated in this tumor sample, and in this mutated gene, what is exactly the protein change. So this protein change here, for example, means in this gene, in this sample, at position 701, and alanine is changed to a valine. So for question 1, you will be asked to count the times a gene is mutated, combining the two subtypes. So you will need to read in all these files and count the frequency of each gene mutation across all the files. And in question 2, you need to count the frequency of each protein change, a gene and protein change pair, and either combining the two subtypes and in each subtype separately. So in question 2, you will have to concatenate the two columns, the gene and the protein change yourself, and count the frequency of gene protein change pair in the two subtypes as a whole and in each subtype separately. And in question 1 and question 2, what we want to see is you report something like a table with one column being the gene name or the gene protein change pair, another column being frequency, actually the occurrence, the count of its occurrence. And in question 2, we want you to compare the gene protein change pair you identified in each subtype separately, and the mutation that is specifically prevalent in one subtype may be the genetic factor that distinguish the two subtypes. So say you see a gene protein pair, a change pair A that happens when counting the two subtypes as a whole, say like 20 times, and you see that this 20 times actually all come from subtype 1, and this particular mutation may be the driving factor for subtype 1. And this is just a clarification on question 1 and question 2. because the results it asks for is not really clear. So for question 1, you need to print out the table of total counts of just gene mutations combining the 2 subtypes. So it's a table with gene name and the count. And in question 2, you need to first get the total counts of gene protein change pair combining the 2 subtypes. And then you do the similar thing, but this time in each subtype separately. So in question 2, we want to see three tables of frequency. And there are some coding tips. You are welcome to use Python if you are familiar with it, but I here recommend you still do it in R and you use the apply-R package for reading in files. Remember, we provide the MA file for each sample separately. So here I present a convenient way to reading all the files together and combine it to a large data frame. So you want to use the dplyr function here. It takes a vector and a function, and basically it will apply this function to each element of the vector, and the results will be stored in a data frame. So you may want to get a vector containing a list of file names for the MAF files, and then you put a function to reading the files. And so in this way, you get a data frame that stores the gene mutations and protein changes across all samples. And to count frequency, I recommend using the dplyr pipeline. So you can give it a data frame and then use this symbol, which means the output of whatever command here is used as the input for the next command. In this way, it saves you a lot of intermediate variables. Say you get a data frame, you want to count the frequency of values in column A. You need to first group by the column, and then you summarize it. You count it using the summarize function, and to count frequency, you use n, and you can store the frequency you got in a new column called count. And you can further arrange the frequency by descending order. So you know the top ones are the most frequent values. And this screenshot shows an example of using this pipeline. So I use the data, the lung disease data from survival package, and I want to know the frequency of values in the ph.column column. So I group by it and summarize it and arrange it by descending order. And so I know that, for example, in this column, the value 90 appears 74 times and 80 appears 67 times, and so on. And you can count the gene mutations, the frequency of gene mutations or gene protein change pair in a similar way. And in the next 3 questions, question 3 to question 5 in part 3, we focus on studying the the tumor driver genes. So this is some background information. So imagine, like, this is an analogy. Imagine like your cell is like this car here, and on the left side is progression to cancer. Normally, you have in your cells proto-oncogenes and tumor suppressor genes. So proto-oncogenes are usually genes that promote cell growth. So it's like the gas for the car. And tumor suppressor gene is genes that stops over proliferation. So it's like brakes. So these 2 types of gene functioning together will ensure that your cells will proliferate and replicate normally, but it will stop at certain time points. So it does not, the proliferation does not lose control. However, in the cancer setting, there are 2 possible mechanisms that could lead to cancer. The first is that there could be some gain of function happening in your proto-oncogenes, and it will become an oncogene. And using this analogy, it's like the gene is overactivated, like your car having too much gas, so you will progress towards cancer. Another mechanism, you lose the brake. That is, it could be some loss of function mutations happening in a tumor suppressor gene. So you lose those checks on your cell cycle, and without a powerful brake here, you can also progress to, your cells may also progress to cancer. So for question 3, you will ask to identify cancer driver genes in glioblastoma. So you will go to this website, and you will choose subset to glioblastoma. I mean, subset studies that are related to glioblastoma. And then you should choose studies you are interested in, and you should also submit the top genes you get from the question 1. And then you look at the positions of mutations, determine whether a gene is likely to be a driver gene. So here is a sample view of invasive breast carcinoma, and I checked 4 genes. And on the very top, it's each color represents your study of origin, and down here you get the mutation of the mutation across these genes. For example, for BRCA1, we see it has a lot of amplification, also have deep deletion, and similar is true for BRCA2. And there is no like quantitative way to determine which gene is likely to be the driver. But if you get multiple genes, you should look at those with many mutations, or they have mutations across a large section of their gene body. So you can identify which ones are likely to be the driver gene. And in question 4, you will be asked to identify whether a mutation is gain of function mutation or loss of function mutation. So you can click into that mutation and see whether it happens frequently at a specific position, or it happens like at very low frequency across the gene body. So for gain of function mutation is usually a single a single mutation that happens frequently at a very specific position, because for gain of function, you may need to change a very specific protein structure at a certain location, changing a certain, say, amino acid. For loss of function mutation, it could happen at many places of the gene body. So leading to change in many, could be a very random place, position in the protein. And this change will very likely lead to loss of function of the protein product. So for loss of function mutation, it may happen across many spots with low frequency. And for question 5, it's very easy. It asks you to look to determine whether patients with a mutation have better survival compared to patients without the survival. So you just need to look at the survival curve, and it will generate for you in this website, and take a look at its log rank test p-values to see if the two survival curves with the mutation or without the mutation are significantly different. And for question 6, it's also very easy. You just it asks you to count the number of clinical trials targeting a certain mutation. So you just go to this clinicaltrials.gov website, search by the disease name, like glioblastoma, and the gene name, and and it will print out the clinical trials targeting this gene in glioblastoma. And there are two ways. You can export a file as a TSV document and read it in R and count using code. Or if, say, there are just 10 trials, maybe you just manually count on the website for those that are still, I think the question is, they are still recruiting people. So it's going to be very easy. Begin. So let's move on to the next part, part 4. And then we focus on CRISPR screens. So the CRISPR systems are originally adaptive immune system in bacteria that they use as a defense against viral genes. But scientists have modified it for genome editing. So the components of CRISPR system include mainly the guidance RNA, the sgRNA that recognize a specific position in the genome, and the Cas protein which is an endonuclease that cleaves the sequence at that position. And we can adopt this system to perform gene knockout. So we design sgRNA or guidance RNA that targets a specific gene, and this Cas9 endonuclease will cleave that gene. So in this way, we can perform gene knockout. This is a brief overview of CRISPR screen. So we first construct a sgRNA guidance RNA library, which contains many different types of RNA targeting many different genes. And we transduce cells with the sgRNA library, usually through the use of viral vectors. And since sgRNA targets many different genes, they can perform genome-wide knockouts. And based on cell survival, we can see positive or negative selection of genes. So positive selection means that a knockout of a gene leads to cell survival. That is, cell expressing sgRNAs for these genes will be actually enriched. And we can use positive selection to find, for example, drug resistance mechanism. For example, after transduction, we apply to the cell culture a drug treatment or a drug treatment, and we will see if under this drug treatment, certain genes with cells with certain genes be knocked out are preferentially enriched, and it will suggest that these genes may be related to drug resistance mechanism. Yeah. And for negative selection, inactive selection, knockout of genes will lead to cell death. So that means cells expressing sgRNAs for these genes will be actually depleted. And clearly this can be used to find survival essential genes. Like if it is knocked out, the cell will die. This indicates that these genes may be crucial for cell survival. And in this part, you will be asked to analyze CRISPR screen data, focusing on positive selection and negative selection of genes with MAGIC, which is a powerful bioinformatic tool developed by Dr. Shirley Liu's lab. And for your convenience, we have already installed the MAGIC environment on Canon for you. So you need to first load Anaconda and source activate this magic environment, and you can use this command magic help to see help documentation for its command. And the CRISPR data is stored at this position. So remember that last time I think someone accidentally deleted the master environment. And this time we make sure that these data files and the environment are locked to students, so students won't have writing permission or editing permission to these files. But when you run any code, do check that you don't write any of your results or change any part of the files in the original data file or the magic environment. So for question 4, it will at the start of part 4. At the start of part 4, it will ask you like to look at the QC metrics of magic, but really you need to first run magic and generate the files yourself. So we provide you the CRISPR screen data at day 0 and day 23, and you need to first generate the count table for sgRNA. So this is the command you will be using, magic count. You need to give it a library file that contains the sgRNA library information, including its ID, the sgRNA sequence, and the gene it is targeting. But we will provide this in the CRISPR data directory. You will also need to specify a prefix for your output file. Here I just use out, but you can choose anything you like. And you need to give it sample label indicating like what are the samples you have in your raw FASTQ files, and then you will provide a FASTQ file. Do note that for the FASTQ files, replicates under a same condition are separated by comma. So we actually have 4 FASTQ files provided, and the 2 replicates for day 0 and 2 replicates by day 23, and they are separated by a comma. But for samples under different conditions, like day 0 versus day 23, they should be separated by a space. So you will get a count.txt file as the output using this command. And here, this is an example I grabbed from the magic tutorial page. And it has columns of sgRNA, the gene it targets, and the sample names. And in the columns of each sample, it contains the count for the sgRNA. And to test if genes are positively or negatively selected across conditions, you should use this command, magic test, and give it the count txt file you got here, and then specify the direction of comparison. Here we like T is like treatment, C is like control. So we compare the CRISPR data from day 23 to day 0, and the prefix is specified using -n, and you can choose whatever you want. And do remember that the labels you use in the magic test command should match those where you are running the count command. That is, the labels here should be the same as the labels in the columns here. And you should consult this page to get an idea of what each file in the output means. And here is some sample script, bash script that you probably want to use to run this command, because these two combined, I think when I test it, lasts for 1 to 2 hours. So you don't want to weighted using the interactive command, but you should submit it as a bash script. And this is some recommended option parameters. And do remember to load Anaconda and the image environment first and put your code here. So finally, to question 1, we ask you to look at the QC metrics. So after running the above commands, you will get QC metrics stored in this count summary.txt file. And you should check this page for QC guidance. It contains information of what each column corresponds to which QC metric. And you will be asked to check whether the percentage of mapped reads is about 0.6, whether the zero count is less than 0.1, and whether the Gini index is less than 0.1. So we give you 4 samples, and if these are true for all samples, and the data is of good quality. And another layer of QC metrics is ribosome genes. And ribosome genes are vital for survival. So knockout of ribosome genes will result in cell death. In other words, they are usually used as negative controls. So the gene summary.txt file contains genes ranked by how negatively they are selected. And since ribosomes are vital for survival, they should be ranked high as very strongly negatively selected genes. But the question is how to select rows corresponding to ribosome genes. A hint here I will provide is that ribosome genes start with RP in their names. So you might want to take a look at the documentation for this function grep and figure out how you subset strings with a certain pattern, like starts with RP. So you want to probably subset to row names that all starts with RP. Yeah, and the third layer of checking QC is to check replicate consistency. So we want to see that the replicates for day 0, for example, are quite consistent. The results we got are quite consistent. So here's a question for you to think about, and this is very vital for this part, is how do we count each replicate separately? A hint is that think of what I talk about here, that replicates are separated by comma. What if we separate each replicate by a space? So treat it just like they are samples under different conditions. And in this question, you will get a count normalized txt file, which contain 1 column for each of the 4 samples. And you want to check replicate consistency by plot replicate 1 versus replicate 2 and check their correlation. For example, for the data, the file here you check the correlation between replicate 1 and replicate 2 for day 0 and replicate 23. Replicate 1 and replicate 2 for day 23. And even easier approach is to plot the heat map of the correlation. And you want to see that the 2 replicates under the same day should be very highly correlated. And for question 2, we ask you to subset those positive and negatively selected genes and figure out what pathways they are enriched in. So in this time, you want to use this gene summary.txt file, and you should read it with a function here, read RA. and you will contain a column for score and another column for FDR. And you use score to determine the direction of selection and FDR below 0.05 to determine the significance. And after you get the positively and negative selected genes, you just run Bayes or gene set enrichment analysis, just like you did in homework 2 to get enriched pathways. And then in question 3, we ask you to find the possible drug targets among the negatively selected genes. As you know, negatively selected genes are vital for glioblastoma cancer cell survival, so they can potentially be drug targets. But here's a problem. What if these genes are also negatively selected for normal cells? That is, to give you a simple example, if we knock out the gene that is essential for energy production in cells, we can definitely kill cancer cells, but it will also kill normal cells. So we really need to consider collateral damage when we choose drug targets. One way to see if a gene is important for both cancer cells and normal cells is to visualize their expression versus dependency across many cell lines. So the more negative the dependency is, the more depleted it is in a gene is in CRISPR screen. So you will go to a website and check for the potential drug targets, their expression versus dependency. So if you see that its expression is highly correlated with dependency, which means that the alteration of the gene expression will affect the survival of many cells, then this gene cannot be used as a drug target. You really want to target those genes that have very weak or no correlation between expression and dependency. And a note here is that sometimes you may not find the genes you get as the top, negatively select genes on the website, but just try using its alias. Just maybe search Google it or search in NCBI gene database to find the other names for the same gene. And in question 4, it's very easy. We want you to identify druggable genes based on public data set. And first, we give you a pan essential.txt file that contains genes that are essential for all cells. And you want to remove those genes from the negatively select genes you got in question 2. And then get the top genes by sorting FDR. And you should go to this Oasis genomics.org and select glioblastoma and input the top 10 genes. And it will tell you which gene is druggable. But when I tested the homework 6, it seems that it's possible we cannot open this website for some unknown reason. So I found another website for you to try this one. And the similar idea, you should go to that website and input the genes. I think in this case, you cannot select glioblastoma, but in any way, just input the gene and see if it will show any druggable information. So for question I think question 4 is the end of part 4. Yeah. So let's move on to part 5, the last part of homework 6, which focus on cancer immunology and immunotherapy. So first, some background knowledge. Tumors tumors do not exist alone in your body. It actually exists in an environment that is infiltrated by many types of immune cells, mainly lymphocytes or myeloid cells, and we call this environment tumor microenvironment. And the tumor infiltrating immune cells will try to eliminate the tumor cells. Uh, for example, the tumors on the surface of tumor cells, you will have, let's say, a complex that will present antigen to, say, an immune cell type like T cells. And T cells, upon recognition of this antigen, will try to eliminate the tumor cells. But we usually find that the tumor cells are not just going to sit there for being killed. It will suppress the function of immune cells through a mechanism called immune checkpoints. For example, it will on the tumor cell, you will have, say, PD-L1 ligand, which can bind to PD-1 molecule on the surface of T cells. And upon recognition, it will suppress the function of T cells. So to knowing this mechanism, many drugs called immune checkpoint inhibitors have been developed to prevent tumor suppression. So we can give them a man-made ligand for PD-1 or for PD-L1. So you will occupy the binding positions so that PD-L1 and PD-1 be cannot recognize each other since their binding site is already occupied by other ligands. And in this way, the function of T cells can be recovered and reactivated to kill the tumor cells. So for this part, I think you don't need any coding, but you need to do some exploration using timer to answer a question that why the immune checkpoint inhibitor therapy doesn't work well in glioblastoma. Well, it works quite well in, I think, SKCM. Uh, I forgot what cancer type is. It's maybe a skin cancer. And for question one, you will be asked to compare the infiltration level of CD8 T cells, which are the effector T cells that kills the tumor cell. And you need to select the gene differential expression gene D tab under the exploration tab. And you just input CD8A and CD8B. They are gene markers for CD8 T cells. And you compare the infiltration level in glioblastoma versus SKCM. And in question two, you will be asked to check the association between immune checkpoint expression and the CD8 T infiltration level in glioblastoma and SKCM. And you need to use gene under the immune tab and just include the CD8 T cells as immune infiltrates. And a note here is that for PD-1, you should really include PD-CD1 because timer uses different name for them. And for PD-1L, it's CD274. And for question three, you will be asked like whether CD8 T cell infiltration affect survival in glioblastoma versus SKCM. And for this question, just use outcome under the immune tab. And last question, I think it's just for a question. It's a question just for graduates. Is that you need to combine your observations and findings from question one to question three, and explain in a short paragraph that why checkpoint inhibitors do not work well in glioblastoma. I think when you get a result from Q1 to Q3, it's very obvious. Yeah, that's the end of the lab.